My name is Katie Fialko, and I am a second-year master's student here at Colorado State University in the graduate degree program in ecology and in the Department of Forest and Rangeland Stewardship. I feel like I've always sort of been interested in ecology and conservation because I was fortunate enough to grow up in the mountains sort of northwest of Fort Collins, so in sort of a rural setting with a lot of space to be outside and just sort of enjoy nature. But with that, there's also always been a lot of problems with people living in the wildland-urban interface, sort of conservation issues that need to be resolved so that people can live outside of towns. And so I think that's what got me into sort of the applied side of conservation and ecology. With my research, I'm trying to determine what drives variability in conifer regeneration in areas where they've done fuels treatments to prevent severe wildfire behavior along Colorado's Front Range. And it's been shown that conifer regeneration is something that can reduce the effectiveness of fuels treatments over time. So my research is looking at what kinds of factors determine why there's a lot of regeneration in some treatments and no regeneration in others. I feel like finishing my field work last summer was really rewarding and exciting because at the beginning of the summer it was still a very daunting prospect. I didn't know how I was going to get it all done. And throughout the summer it was definitely exhausting a lot of the time, but I got to work with some really lovely people who also made it fun, and looking back at the end of the summer, it was definitely very satisfying to see everything that we accomplished.
Good afternoon. I'd like to welcome everyone to the second interdepartmental seminar. I'm Allison Foster. I'm a master's student in the Forest and Rangeland Stewardship Department. I have the pleasure of introducing Katie. We also have a second speaker as well this afternoon. Katie is a master's student in the graduate degree program in ecology, studying under Dr. Seth X. She is studying the long-term effects of preventative fuels treatments in low elevational forests. Katie also holds her undergraduate degree from CSU, which is in natural resource management, which she completed in 2013. In the time in between her undergraduate and graduate degrees, Katie has worked for the USGS here in Fort Collins as a biological science technician and as an education program coordinator for the Sequoia Riverland Trust. Today, Katie will be speaking about her research into the efficacy of fuel reduction treatments in Front Range dry mixed conifer forests to reduce ladder fuels and crown fires. She is excited to be studying a topic that is relevant to the area and that is in hopes that it will help fire managers schedule fuel treatments. With that, please help me join in welcoming Katie.
All right, thank you. I'm Katie. I'm very grateful for the opportunity to come and talk to you about my research here today. My project has been examining conifer regeneration in fuels treatments in Colorado's dry mixed conifer forests. And I am a master's student who started my degree last fall, in the fall of 2016, and so I collected all the data for my project this past summer, and I'm still working through my analysis currently. So this is sort of a research in progress kind of presentation. So with that, I'll go through a little background on forests and fire in Colorado and on the fuels treatments we do to prevent severe wildfires. I'll discuss some previous research very briefly from other FRS department researchers here on conifer regeneration. And then I will go over my research objectives and sampling design and some very preliminary results that I have to share with you. So first, to begin with, when I talk about dry mixed conifer forests, I am talking about lower to mid-elevation ponderosa pine and Douglas fir forests. At higher elevations, these grade into lodgepole pine. And generally, across the Front Range, today these forests are a lot denser than they were historically. And this has a lot to do with past management practices that focused on complete suppression of any and every wildfire on the landscape. So in these photos, these are paired photographs from 1903 on the left and 1999 on the right, so about a century apart. And these were taken on the South Platte River near my study area. And you can see that there's a really dramatic increase in tree density over the course of the past century. This is important because this greater tree density is a large reason why we're seeing a lot more severe wildfire behavior in Colorado. A really great example of this is still the 2002 Hayman fire, which burned over 138,000 acres in the Upper South Platte watershed, where I did my study. And obviously there's been a lot of other really large, severe fires that we've seen since this as well in Colorado and throughout the West. And these wildfires do a lot of damage to human property and to watersheds. They have negative impacts on water quality downstream. And another interesting impact are the long-term implications for conifer regeneration in these large burns. Marin Chambers, who works for the Colorado Forest Restoration Institute, studied conifer regeneration in the Hayman burn area, and she found that 70% of the severely burned area from this wildfire exceeded the dispersal distance for seeds from surviving trees in the burn. And so what this means is that there's a huge area in this fire that's not going to be forested again for many, many years to come. So what forest managers are trying to do is develop fuels treatments that can prevent severe wildfire behavior. And these may be things like thinning or chipping or mastication of fuels, or prescribed burning, or some combination of the above. And it's pretty well established that these are effective in the short term for a couple years after treatment. So these photos were taken following the 2002 Cone Fire in California, and they were taken back to back on a treatment boundary. So on the left, we're looking into a treated area that was thinned and then followed up with prescribed burning. And on the right, we have an area that was untreated. And so you can see there's a really big difference in overstory mortality between the treated and untreated areas. So the treatments work in the short term, but a lot remains to be seen, a lot remains to be understood about how they work in the long term as well. And conifer regeneration has a lot to do with this. One of the main principles of reducing fire hazard is to reduce surface fire behavior in order to limit the potential for crown fire. And conifer regeneration contributes to surface fire behavior by serving as a ladder fuel to transfer fire from the surface into the crowns of trees. And it also can grow up to contribute to future stand density and make it easier for fire to transfer between the crowns of trees. Crown fire is something that should be avoided because it's what results in high mortality among trees during wildfires. Researchers from this department, again, including my advisor and several others, published a paper in 2016 where they were able to link, using modeling, they were able to link the densities at which conifer regeneration begins to reduce treatment longevity. So we have that connection, and what remaining research needs to do is to help understand what drives variation in conifer regeneration in these treatments. And so previous research has addressed this question. In a study done by another former graduate from this department, David Francis, he looked at conifer regeneration in thinning and thinning followed by prescribed burning treatments in the Arapahoe-Roosevelt National Forest, just west of town up near Red Feather. And he looked at treatments ranging in age from 3 to 12 years old. And some of his key findings were that there was no real difference in regeneration densities between the two treatment types that he looked at. So that's pretty important. And he also observed a really high variability in regeneration densities between the stands that he examined. And along with this, he observed that in general, north aspects had twice the regeneration 10 years post-treatment as south-facing aspects, and that this was largely due to the presence of Douglas fir. Douglas fir is a more shade-tolerant species than ponderosa pine, so it likes the cooler, wetter conditions on north aspects and regenerates more readily there. And then the final piece to this was that Douglas fir regeneration was highly correlated with the amount of Douglas fir left over in the overstory post-treatment. So my research follows up on this previous work by David Francis with some important differences. I'm sampling in a different part of the Colorado Front Range. All of my work is much further south. And I'm also sampling across broader elevational gradient. So I actually get into a forest type that includes a little bit more lodgepole pine in addition to ponderosa pine and Douglas fir. And I'm also comparing different treatment types. And then lastly and importantly, I have ages for all of the seedlings that I collected over the course of the summer. So this means that I'll have very accurate estimates of regeneration in treatments that was there before the treatment versus what came into the treatment after they thinned or masticated. So my research objectives were first to just characterize the quantity and species composition of conifer regeneration in treatment areas and look for general trends and patterns. And then second is to develop sort of a predictive model of conifer regeneration in treatments that could hopefully be of use to managers who need more information about when to go back into areas to retreat due to seedlings. And then lastly, I was also fortunate enough to be able to work with the Colorado Forest Restoration Institute this past summer while I was collecting my thesis data to also collect data for their fuels treatment monitoring program. And this was actually part of the Forest to Faucets initiative that Joni Chang spoke a little bit about last week. So to that end, I focused on sampling and treatments that were intended to prevent severe wildfire in Denver's watershed. And so these were on Denver water and National Forest Service lands in both the South Platte and the Boulder Ranger districts. And the rough locations of these are here. I'm sorry my map isn't very pretty. But so I sampled up in Boulder. You can see Highway 119 kind of heading west out of Boulder, and 72 going north between Nederland and Ward, and those were where I put in higher elevation plots that had more of a lodgepole component. And then in the South Platte, where I did the bulk of my sampling, you can see 285 coming down here out of Denver, and then this is the 1996 Buffalo Creek burn scar here, and then this is the Hayman burn down here. So just to give you sort of an idea of where all my my treatments were located. So in total, I sampled in 229 plots, and these were in a variety of treatments ranging in age from 5 to 14 years. In the South Platte, I sampled in both mastication and thinning treatment types. And in the Boulder Ranger District, I only sampled in thinning treatments. This is because there ended up not being a comparable mastication treatment type for me to examine in Boulder. So I'll use these plots to just look very generally at how conifer regeneration responds in treatments in general. The plots ranged in elevation from just over 6,000 feet to just over 9,000 feet. And I also put all my plots in on aspects within 45 degrees of north or south. And so elevation and aspect are very important for regeneration because they are a large part of what dictates the temperature and moisture conditions for seedlings coming in. So I made sure to sort of evenly distribute all my plots across combinations of treatment type, elevation, and aspect to capture that full range. So, preliminary results. I collected in the end close to 1,800 conifer seedlings, along with their species and height. And then this summer, or this fall, I had some very lovely technicians who helped with aging all of these seedlings, which was a big job, so thank you. And we found that they ranged in age from 1 to just under 100 years. So it's a little misleading to call these seedlings because they're not really, a lot of them aren't really small trees that are going to grow up into big trees. A lot of them are just small trees that are probably going to be small their whole life. With that, using these ages, an important finding was that a little over 60% of the total regeneration I sampled was actually what's considered advanced regeneration. And so these were seedlings, these were small trees that were present in the plots before the time of treatment. This is kind of interesting because if we look at just the total regeneration numbers from all my plots by treatment type, there's not a really big difference between the average trees per hectare in mastication treatments versus in thinning treatments. But if we split it apart and subset just that post-regeneration, post-treatment regeneration coming out, there appears to be a little bit more of a difference. However, I haven't actually analyzed this, statistically analyzed this yet. So whether or not this is significant remains to be seen, especially when a lot of other variables are considered as well. Similar to what David Francis found in his work, there was a very wide range in total density of seedlings. We had a lot of plots with 0 seedlings with no regeneration, up to plots with close to 11,000 seedlings per hectare. And then we also had 1 or 2 plots with even more seedlings. One plot in particular, scaled up, had close to 45,000 trees per hectare. This is probably going to end up being considered an outlier, but I still think it's important to note that there are conditions like this out there where there's just a lot of small trees. I included this plot photo in particular because you can see where the quadrant is where our plot center was, and it would be around there where I collected seedlings from. But a little bit further down, you can see in this green here, that's like a big clump of Douglas fir seedlings. So on the same north-facing aspect within like 20 feet of each other, there's, you know, a really broad, a really big difference in regeneration density. And also along with that, as I said, this is on a north aspect, and the variability in regeneration density that I've observed was much broader on north aspects than on south-facing aspects. We saw a lot of plots on both aspects where there weren't any seedlings, but we really only saw the high density, the super high density regeneration condition on, in plots on north-facing aspects. And then with regards to species composition, I observed a much higher mean density of Douglas fir as opposed to ponderosa pine. These numbers in general were a lot higher than anything reported by David Francis, so that's of interest. Probably something to do with being geographically further south along the Front Range in Colorado. There's different precipitation patterns that might result in more favorable regeneration conditions, those kinds of things. But again, Douglas fir, mean Douglas fir density was a lot higher than ponderosa pine. And I've also, in just preliminary results, observed that Douglas fir regeneration does appear to be correlated with the residual density of Douglas fir trees in the overstory. And this is just using really basic linear pairwise correlations. So again, further statistical testing needed to confirm what I'm seeing. But this does not appear to be the case for ponderosa pine. So far, ponderosa pine doesn't appear to be very strongly correlated with any of the numerical predictors that I'm including. But the strongest one does appear to just be time since treatments. So perhaps ponderosa pine seedlings are accumulating in treatments over the course of the treatment lifetime. And so lastly, obviously I have a lot of next steps because I've just begun my analysis. And one of the first things I need to do is to finish preparing a little more data I have that I'd like to add to the variables that I'm using to predict regeneration density. The first of these things is site productivity. So in each of my plots, we also collected a tree core from a site tree, where we'll have the height and age of one of the most dominant, best looking free to grow trees from the plot. And this will be used to determine site index, which is an indicator of site productivity. And it's something that can be compared across plots. So I'll add that in there. And then lastly, we also collected a lot of understory data as a part of CFRI's monitoring protocols. And I'm hoping to use this data on shrubs and herbaceous understory vegetation to determine plant associations for each of my plots. Plant associations just consist of the dominant overstory tree as well as understory plant species in an area. And they're interesting because they sort of reflect elevation, aspect, topography, soils, all those things all at once, and they might actually be a better predictor of seedling density than each of those abiotic factors alone. And if they are, they could be a really simple and easy to use tool for managers in predicting how long their field treatments are going to last. So once I finish preparing the rest of my data, I'll work on my actual analysis, where I'll use mixed regression modeling to more formally examine the relationships between conifer density and all the variables that I've talked about today. And hopefully these results can be used to help inform management to make better decisions about when they need to go back in and retreat fuels treatments. And so with that, I have more people to thank for all the help I've had on my project than I can list up here or name. But thank you, and if you have any questions, happy to answer them.
How are you defining seedlings and saplings?
Yeah, so seedlings are any trees less than DBH, so less than about 4.5 feet tall. So as I mentioned, a lot of trees that are less than this tall are still very old. They're not necessarily young trees.
So the 96-year-old tree was less than 4 feet.
Mm-hmm. Mm-hmm. Indeed.
Based on your observations in the field, do you have a sense of whether or not the habitat type element is going to play out? Or it's going to look any different?
I noticed some differences in certain areas, and in some areas maybe less so. I'm not sure yet. Like I said, in a subset of plots, it seemed like there were some really strong things coming through, but in other areas it was all just noise. I don't know.
Are you seeing a consistent age for a certain size with each of these species, or is there a lot of variability? So like if I saw a 3-foot tall ponderosa pine, is there that's a 18-year-old ponderosa pine, or is there a lot of variability?
I think there's a lot of variability, just depending on how suppressed certain seedlings might be. But I'm not. There might be some trends there. I'm not sure.
Do you think you'll be able to compare the advanced regen and the more recent post-treatment regen composition? I'm curious if individual treatments might change the trajectory of which species respond, not just how many, but. Yeah, I am definitely going to use those two groups differently in my analysis. One of the things that I'm trying to resolve right now is the fact that there's just a lot of very small seedlings in certain treatments, and I know that they're not all going to survive. So there might be some potential to use the advanced regeneration numbers and ages that I have to sort of estimate how many of the smaller seedlings are actually going to make it and become something that reduces the treatment effectiveness. But yeah, comparing those two groups differently is something that's definitely going to happen.
Did you go all the way down to like 1-year seedlings?
Yeah, I did not count like seedlings from this year. They all had to at least be a year old with hardened stems, and we did observe a lot of very, very young germinants out in the field, especially early on too, though.
Mhm.
Is there any pretreatment data?
There is not. I focused on sampling only in treated areas, so my study only looks at, there's no kind of comparison between a control and treatments. It's just focused on treatments in particular. Mm-hmm. Mm-hmm.
Okay.
Yeah, sorry.
I could ask you 1,000 questions. In terms of longevity, what metric are you gonna use in terms of deciding how long a treatment is needed and subjective?
My project is mainly just focusing on the density of seedlings, and as I mentioned, in the modeling paper that I mentioned earlier, there has been research that's tied specific densities of seedlings to so many years, reduction in treatment longevity. So I'm focusing more just on the seedling density aspect. Mm-hmm.
Well, with that, let's say thank you to Katie.